welcome my colleagues, who I am so honored to be part of their team. These guys and gals are the world's leading subject matter experts on perennial grains and ecological intensification, and our Director of Research, Tim Cruz, is going to get them started. Thanks.
Thank you. So a literal warm welcome to all of you friends of the land sitting here next to the Smoky Hill River today. My name is Tim Cruz, and I serve as the research director and an ecologist here at the Land Institute. It's my pleasure to orchestrate this research update in which some 10 of us, or 12 maybe, are going to offer you a few highlights of the last year. And there are a lot of highlights. It's difficult to choose. From sorghum genetics to soil microbiology to an expanding research arrangement with colleagues in China, research to create agroecosystems that function more like the natural ecosystems they replace continues to progress and expand. Let me start with China. In the first half of 2017, we had not one, but two delegations visit the Land Institute from Yunnan University and the city of Kunming, where Yunnan University is located. These delegations helped build support in China for the creation of a perennial grains center at Yunnan University, which in fact did come to fruition in June during an international perennial rice meeting in Kunming that I attended along with our perennial wheat breeder, Shuwen Wang. At the meeting in June, TLI and Yunnan University signed a joint project agreement that outlines ways in which we can expand our collaboration and cooperation. Two more groups of Chinese research visitors will be coming to TLI at the end of October, and we will be sending David Van Tassel and Fred Yutzi to Kunming for a perennial rice workshop around the same time. That was highlight number one. Specific to ecology, another international effort we have become involved in is the University of Copenhagen's Deep Frontier project. Led by Christian Thorpe Christensen, a team of ecologists, soil and crop scientists, and microbiologists have taken a deep interest in deep-rooted crop species. Specifically, they are wondering, what are those roots actually doing down there? Collecting nutrients, water, sequestering carbon, cultivating strange microbes. To help answer these questions, they have installed dozens of what are called mini rhizotrons, which are essentially clear tubes installed in the soil so you can drop a digital camera down and examine what roots and other visible organisms are doing at different soil depths year-round. They have also built above-ground towers that are 4 meters, or about 12 feet tall, to observe and sample different plants, roots, and rooting zones. Among a small list of deep-rooted species they are studying intensively are our own silphium oilseed crop, kernza, and alfalfa. There's fascinating ecological research taking place in Kansas as well, and the next two folks up will be describing some of it. A project I wanted to mention is the establishment of the first commercial perennial biculture plots. Up until now, kernza that has been grown for commercial applications has been grown in single species stands, often organically, but using manures as nutrient sources. This summer, the leadership of Patagonia Provisions indicated that they were interested in taking kernza production beyond minimal organic certification or compliance to the most regenerative level possible today. After some back and forth, we agreed that sowing 10 to 12 acres of perennial legume and kernza intercrops at the Perennial Agriculture Field Station in Lawrence, Kansas, and at the University of Wisconsin, or at a farmer's field near the University of Wisconsin under the supervision of Professor Valentin Picasso, who is here today, somewhere. He's here. Okay. So we're a little nervous but excited that the first multi-species perennial grain fields are being sown this fall to see what biculture production involves at the field scale. That's a big deal. All right, okay. I will pause. Thank you. If there's one topic in ecology research that crescendoed at the Land Institute since last Prairie Festival, it is soil carbon. It is a topic that has experienced something of an upwelling in the consciousness of a particular faction of society. And something tells me that some of this faction might be present in this barn. Who, for example, has thought for 5 minutes or more about soil organic carbon this last year? Okay. Who spent 5 minutes thinking about soil organic carbon 5 years ago? Okay, that's okay. Case rested. A lot of people are thinking more about carbon dioxide concentrations in the atmosphere and how humanity has influenced these concentrations. I am one of these people. And I think I can speak for most of us in expressing frustration at industrial society's unwillingness to seriously address the most important source of human-caused atmospheric CO2 increases, which is the combustion of fossil fuels. Given the lack of serious progress in curbing fossil fuel emissions, people have begun to look with understandable urgency for other ways to reduce atmospheric CO2, and many eyes have ended up looking straight down at the ground. You see, soils hold in organic forms between 2 and 3 times the carbon that is contained in the atmosphere. And a recent paper in the Proceedings of the National Academy of Sciences suggests that human activities, namely crop agriculture and livestock grazing, have been responsible for reducing soil organic carbon levels by a total of 8% globally. That's 8% of all soil organic matter on the planet. The lands that have experienced the largest decreases in soil organic carbon are plowed annual croplands, which have lost between 20 and 70% of soil organic carbon. We will demonstrate why in a second. Some of you might be asking, what is the difference between soil organic carbon and soil organic matter? Carbon is just a major ingredient in soil organic matter, making up about 58% of it, with the rest being oxygen and nitrogen and hydrogen and some other nutrients. Given this situation, interest in how people could possibly recapture that 8% of soil organic carbon has exploded. And we have been pulled into this conversation because what we are working on at the Land Institute, the development of perennial polycultures, is among the most robust, if not the most robust approach for achieving soil carbon resequestration. The bad news, or at least an important reality check, is that soil carbon resequestration is not going to eliminate the need to address fossil fuel emissions. Far from it. If we were able to recapture all the soil organic carbon that has been lost from croplands, it would offset about 3.5 years of fossil fuel emissions at 2014 rates. If you add all other sources of soil carbon loss, like degraded rangelands, the offset would go up to about 8 years' worth of fossil fuel emissions. Now, the good news is soil organic carbon sequestration is not insignificant. Every carbon sink matters. And this one in particular is desirable because aside from pulling CO2 out of the atmosphere, enhancement of soil organic matter benefits many aspects of crop production. It is, in fact, the holy grail of ecological intensification. Sir Robert Howard to Ruth Stout to Ken Kasman all highlight the importance of soil organic matter to regulate the supply of nutrients to plants, to help hold soil moisture, to maintain good soil structure, which helps with things like water infiltration, and even to promote disease resistance. So why do we think our work runs ahead of other proposed approaches to recapturing the soil carbon that has been lost from croplands over the last 10,000 years or so? To answer, I'm going to ask one of our incredible interns, Nadine, to approach the stage and help me with an exercise that I really hope works here. Okay, I'm going to pull this back so folks over here can see. Okay, Nadine, if you want to take here. So here's yours. Okay, so imagine that this is the soil organic matter in a healthy prairie soil, okay? And what we're going to walk you through is turning the prairie into annual agriculture and then into a perennial polyculture with respect to this soil organic matter, okay? Now, soil organic matter is not a static property. It is a balance between inputs and outputs. Inputs primarily being roots that send photosynthate down into the ground, sugars and carbon down into the ground, and those roots turn over, they die, and they slough off, and that feeds the creation of soil organic matter. Outputs are microbes. Whoops. Microbes eat the soil organic matter and breathe out carbon dioxide. And so it's through CO2 that the balance is struck. Now, microbes can really chow through organic matter, and with the right organic matter quality, oxygen, water content, it'll just disappear quickly, and that's what's called a compost pile, okay? But soil has silt and clay that bind to soil organic matter, form aggregates, and protect it to some extent from microbes. At least it slows microbial consumption of organic matter down quite a bit. So that's why there's this lag, and that is the soil organic matter pool, okay? So a healthy prairie has a pretty large amount of organic matter input from roots. If you want to go ahead and add it to the pool, this is the annual addition of soil organic matter. And meanwhile, the microbes are respiring out here, okay? This is going on all the time out in the prairie. Okay, thank you. And guess what? Soil organic matter is basically the same. So we're not really growing it or shrinking it. It's more or less in an equilibrium. Okay, so now we come along and we plow this field and we plant annual crops. Both the inputs and the outputs are rather dramatically changed. Here, I got this. This is yours. Here you go. So annual plants only allocate about 15 to 30% of their productivity below ground, whereas perennials allocate 50 to 60% of their productivity below ground. So the inputs of soil organic matter decline substantially, and guess what? When you bust those clods open with the plow, it opens up this organic matter that is now available to microbes, and they just have a field day. And so we have a lot, go ahead, we have a lot of microbial respiration happening after the prairie is first plowed. Oh my. Oh yeah. Okay. This is good. Now I need to get one in here. Okay, so here, go ahead and don't let me forget this. Okay. Okay, now. Okay, so you see what happens? We've had a significant decline in soil organic matter. And in the first year, you have, our first 5 years, you have this rate of decline. But eventually, that newly exposed soil organic matter gets consumed by microbes, and the respiration levels begin to decline in organic systems. So go ahead and pour that in. But still, you have more respiration of carbon being lost from the soil than you have from these measly annual crop root inputs. Okay. All right. And so eventually, finally, you reach a stasis, even in organics, in annual crop systems, that is a reduced equilibrium. Go ahead and pour it in. Where once again, you have no net change in soil organic matter year after year, more or less. I mean, it goes up and down depending on the climate and things like this. Okay, so that's where most annual croplands are right now. And the big interest in resequestering soil organic matter is in bringing this back up and recapturing that organic matter. And so with a perennial polyculture, when we plant perennial polycultures, we address both the inputs and outputs. We have more carbon going into the soil than we have right now with our annual crops. Go ahead, right now, and pour that in. But we still have reduced respiration because the microbes are actually not able to get at a lot of this carbon since it's being tied up in these new aggregates. We're not disturbing the soil, and so we have increased soil organic matter. Okay, let's go ahead and do that again. So bear with us when we do this one more time to bring it to a beautiful completion. I mean, in reality, this is probably where it stops, because we probably are not going to achieve the level of soil organic matter that the native ecosystem have for a lot of reasons. But for the sake of a beautiful demonstration, we will go ahead and bring it back. Okay, so there you go. How about it? So hopefully that gives you a sense of why what we're doing will help address the issue of resequestration. And if you start playing it in through your head, why other approaches that people have to achieving resequestration, most of the methods address either the inputs or the outputs. No-till addresses the respiration losses. Addition of cover crops addresses the inputs, but only the cessation of cultivation or tillage and the replacement of perennial roots in the place of annual roots will actually approach what the prairie originally had in soil organic matter. So we're going to have questions at the end if there's time, and if any of you have questions, we also have the plot tours following this and dinner, but we do have to move on. And it's my pleasure to introduce two colleagues, two postdocs, who are at the University of Kansas right now. Liz Kojal is a postdoc in Jim Beaver's lab, and she's working on mycorrhizal fungi in our systems and in analog prairie systems. And Liz is going to tell us a few things about what she's doing. So give a round of applause for Liz.
Hello, everyone. Thanks for coming to my first Prairie Festival. So I'm Dr. Liz Kojal, and I'm a postdoc at the University of Kansas, and I work with Dr. Tim Cruz with Perennial Polycultures at the Gorell Farm field site that was just initiated in Lawrence, Kansas. And so I'd like to start talking to you all today about what I did before I came to the Land Institute. So my background is in prairie restoration work and with mycorrhizal fungi. I study arbuscular mycorrhizal fungi, which are below ground soil microbes. They aren't like the fungi that you eat. They're microscopic, and they spend their entire life cycles below ground. So although they're small and you can't eat them, we do eat a lot of plants that depend on these mycorrhizae. About 80% of plant species associate with these fungi in a symbiotic relationship. The basic symbiosis is that fungi explore the soil and collect nutrients that they deliver to their plant hosts, and in exchange, plants release sugars from the roots, which feed the fungi for their hard work. And so with mycorrhizae's help, plants can have greater access to soil nutrients and water and can become more productive. And so it's this mutual exchange that keeps the symbiosis going between plants and below ground soil fungi. So as a PhD student, my work in the prairie system compared mycorrhizal communities in old-growth, pristine prairies relative to those that are found in disturbed soils, such as agricultural fields, or in prairie restorations, which often happen in post-agricultural landscapes. And it turns out they're really different. Basically, what we found is that the things we do in conventional agriculture really disturb these below-ground fungi. Tillage, the use of fertilizers, and other chemicals can alter fungal communities and leave them less abundant and even make them less beneficial to their host plants. Additionally, some species of fungi do not reoccur once the soil has been disturbed. And so my research is very applied. My process is to go to old-growth prairies that contain healthy soil fungal communities, and I collect and isolate these prairie mycorrhizal fungi. I can grow them up in the lab, in the greenhouse, and then I can apply these prairie mycorrhizae into prairie restorations. And what I found is that many prairie plants grow 10, even 25 times larger with the addition of these prairie mycorrhizal fungi. They're really dependent on them. So we find that more prairie species are recruited from seed with mycorrhizal additions, which can result in less competition from weedy plants, for instance, that tend to be less reliant on mycorrhizal communities. So my past work in the prairie has a lot to do with the goals of the Land Institute. Many of the prairie species we found to respond most strongly to mycorrhizae are perennial species, such as lead plant, meads, milkweed, culver's root, prairie dropseed, and others. Perennial agricultural plants are similar to these perennial prairie plants we have found to benefit so much from mycorrhizae, and that they both invest a lot of energy to below-ground processes, such as creating a lot of roots and long-term perennial root structures. They tend to grow more slowly and reproduce more slowly than annual plant species. And additionally, they can live a long time. Some of these species can live decades, so they're interacting with the below-ground microbial community for a long time. And some of the species I've worked with in the prairie are even the same as the ones we work with here at the land, such as the oilseed crop Silphium, which I'm sure David is going to talk about, but maybe not, I don't know. So in my first year as a postdoc, I've worked with many of the researchers here on the stage to test how their perennial agricultural crops respond to prairie mycorrhizae applications. And I found that some of the candidate perennial crops are strongly mycorrhizal and benefit from these inoculations in the field. So I'd like to use this information to improve the field establishment and the growth of these perennial agricultural crops in future plantings. And so another thing that I spend a lot of time doing is working on my small business, Michael Bloom. So because of my work in prairies and with both annual and perennial crops suggests that many plants benefit from native fungal communities, especially these prairie fungi that are extremely beneficial. I want to provide the fungi I've collected throughout my research career to the public through Michael Bloom. And so currently I'm writing grants for small business innovation from the USDA and other programs that will help me be able to test if we can get this process of microbial amendments that works really well in small research plots transitioned into this type of scale that can function, for instance, for kerns of plantings. And so that's what I've been up to the last couple of years. Thanks for listening.
Thank you, Liz. I also wanted to say I was so excited that the soil organic matter equilibrium was achieved that I didn't thank Nadine for helping me wherever. Okay, so thanks Nadine for her assistance. Next, Tom McKenna is a postdoc in Ben Sykes Lab at University of Kansas. We're also working on soil microbial topics. And Tom, what do you got going on?
Good afternoon. So the Sykes Lab, in the Sykes Lab, our focus is on microbial ecology with emphasis on soil fungi. And microbes in plant communities are important because they can be plant pathogens, so they can cause disease which affect growth and yield. They can also be mutualists, an example of what like Liz just told us about, so they can form relationships with plants that help them acquire nutrients. And others are saprotrophs, so they play a role in decomposition and nutrient cycling, which affects the availability of nutrients for crops. In perennial agriculture, crops are in the same field for multiple growing seasons without disturbance, so there's more time for the soil community to develop. So there's a type of succession going on in these soils and these fields. However, this means there could be an accumulation of good microbes, like mutualists, or bad microbes, like pathogens. In monocultures of conventional agriculture, farmers typically ameliorate these pathogen problems by using crop rotation. So they either till or herbicide and then plant a different crop, and then you can avoid the pathogens. However, in perennial agriculture, with the absence of crop rotation, there may be a need to plant a combination of species, so increase the diversity in the system to reduce pathogen abundance. So what I'm really interested in is how different perennial crops and different cropping strategies, like the planting of one versus multiple species, whether you till or don't till, shape the soil communities over time, and also how the communities cultivated by different crop communities, how do they influence or mediate subsequent crop growth. So basically, are there more good guys than bad guys over time? How long does it take for these soil communities to form? And also, how do they affect crop yield? So in order to understand what's going on in the soil and see how communities change with differing crop strategies, our lab uses a variety of different methods, including DNA sequencing and plant growth assays. So using DNA extracted from the soil, we can get an idea of who is present in a community, and we can tell the relative differences and abundance of taxa between different cropping strategies. To understand the function of certain soil communities, we typically use somewhat of a black box approach, where we take and grow individuals in soils conditioned by different cropping strategies or different plant species, and then we measure these different responses. So we look at above and below ground biomass and then also disease incidence. And these experiments can be done in the field or in the greenhouse. So right now I'm actually working on some data that was collected from the ART plots or the agroecology research trials, which are just here across the river. These are long-term plots, and we collected soils from the treatments where they had intermediate wheatgrass monocultures, so one perennial species. We also collected soils from plots that were a form of prairie reconstruction, so it contained native prairie species, so that's many perennial species. We also collected from plots that were in an annual crop rotation. So there were 3 different crops planted with tillage in between. So those are monocultures, but they had diversity over time. And they also had tillage, which the other plots did not. Our initial DNA sequencing results showed that fungal communities in the wheatgrass monocultures and the prairie reconstructions are very similar, but both are very different than the annual plots. So this reinforces studies that suggest that tillage is a dominant driver of the soil community within systems. However, I still need to get into the data further to see if there are any other mechanisms driving these responses, because within tillage there are a number of confounding factors such as the addition of nutrients. To understand the function of those soil communities, we took soil from here and took it to Lawrence, and then we planted Kernza in those soils, and then we took assessments of above and below ground biomass. And unfortunately, I don't have data on that yet, so I can't present that today. And then we are also in the process of installing 2 field experiments, one here in the ART plots and then one at the Perennial Agriculture Field Station in Lawrence to further investigate this relationship, and I hope to present those results here in the future.
Thank you, Tom. And now I'm going to ask Lee DeHaan to come up and tell us a few things that have happened with Kernza this year. It's been kind of a big year for Kernza, and so look forward to what Lee has to say.
Yes, as Tim said, we've had a lot of exciting things happen this past year. A lot of that happened around commercialization. I'm actually going to talk about that tomorrow, so I'll defer those comments till tomorrow and keep it very brief this afternoon. First of all, so we all know what we're talking about with Kernza here. This is the grass plant that we're domesticating into a grain crop. It's on the poster over here, the large root system. Also, it was in the pancakes this morning. That was 100% Kernza grain in those pancakes you had this morning. And it seems to be working really well for being able to make things like pancakes, even breads out of it. But back to the science of what we're doing, most of it is genetic improvement. So we're taking a very wild grass that originally came from the Middle East. We're selecting it for larger seed, not to drop its seed on the ground, and higher grain yield, and figuring out how to grow it. And that's kind of the whole domestication process that we're going through. Our current target is really something that's a dual use as a forage crop, where the leaves and stems are fed to livestock, and the grain is used for human consumption. We have research collaborations now around the world and in the U.S., a few places: Sweden, France, Belgium, Canada, Australia, New York, Ohio, Michigan, Wisconsin, Minnesota, Iowa, Kansas, etc. So many collaborations at universities that are helping us to get at the really tricky questions about how to develop a totally new plant from wildness to something that has a high grain yield, to do the kinds of good things like carbon sequestration that Tim demonstrated earlier. Our work is kind of in 4 areas: genetics. We're looking at ecosystem services, which is like increasing soil carbon and measuring that, how much happens, how quickly. We're looking at utilization, so what can it be used in, what kind of products, how long will it last on the shelf, and then management questions and ecology questions, so what's the best way to maintain the yield over time, how do we keep the weeds out, what time do we plant it, what kind of fertility does it need, all these basic questions that we've really had over 100 years of science on our basic crops on. We're starting from scratch and answering all those questions again for this new crop. My work is mostly in the genetics, and I think I'm out of time. So basically, the work over the past few years has led us to have a draft genome for the species, and the genome, or the size of the genetic information, is about 4 times the human genome, a little smaller than wheat. So it's an enormous project. With the newest technology that we could use, we were able to do this in just a couple of years. And quite affordably, but we have many partners at many institutions that are working on doing this kind of mammoth project. And then we're starting to use that information. So we're starting to be able to use that in development of perennial wheat, which Catherine will probably talk about, but also in the domestication to help us accelerate. So by looking at the genes themselves or markers for genes, we're hoping to more than double the progress that we can make in any given year. So going from taking maybe 2 years per generation to 1 year, and even getting more efficient than that. So we're hoping to catch up more quickly. We know we have a long ways to go to get the yield targets that we're looking for, but it's being useful now, and as we increase the yield over the next few years, we hope for even better things to come.
Thanks, Lee. And like Lee said, there will be a panel on Kernza commercialization tomorrow morning. So, and now I'd like to welcome Stan. Are you both coming up, or are you coming up first? Okay, Stan Cox, the sorghum breeder here, and then Fiona, and they have some special guests as well. So, Stan.
Great. Thank you. We think of sorghum, or milo as we call it here, as a Kansas crop because Kansas is the biggest producer among the 50 states of sorghum. But we're really growing an African grain. Sorghum, a wild perennial a wild annual sorghum grass was domesticated in East Africa about 8,000 years ago as a crop, and has been grown on the continent ever since. It came here, was brought here in the 20th century from East Africa and South Africa by USDA and university scientists, and was developed as a major grain crop in this country. Meanwhile, sometime after 1492, the perennial weed, sorghum halepens, or Johnson grass, arrived on its own on the continent as an immigrant seeking a better way of life in the New World, and in which it succeeded. And our perennial sorghum program is based on crosses between these two species, bicolor and halepens. So it was only natural that starting about 4 years ago, we began working with our colleagues in various countries in Africa and Asia on developing perennial sorghum that would be adapted in those very different environments. And we've been fortunate for the past 4 years to be participants in a project funded by the Agency for International Development and led by Andrew Patterson at the University of Georgia, which involves colleagues in all over Africa and in India on developing more drought-resistant sorghum, super ratooning sorghum, and eventually perennial sorghum that's adapted in those countries. And I'd like the folks who are, we've been, some of them are here because this week we get together annually, and this week we were lucky that everybody came here to Salina to meet, and some of them have stayed on for Prairie Festival, and I'd like to introduce them. If y'all could stand up. On the back row there, from left to right, is Dr. Bongani Ndimba from the Agricultural Research Council in the Republic of South Africa. Next to him is Andrew Patterson, University of Georgia, who's our leader. Next to him is Dr. Balua Nebi from Mali in West Africa with the International Crops Research Institute for the Semi-Arid Tropics, or ICRISAT. Is Yana here? Oh, where are you? Sorry. Yana Kolova up in the balcony is also with ICRISAT from, based in India. And then down here in front, we have Eric Manyasa from ICRISAT in Kenya, and Wen Chen Kong, who works at the University of Georgia with Dr. Patterson. And I'd like to, I wish I could get everybody to say something, but I had to just settle on a couple of people. And Eric has grown Land Institute perennial sorghum material in Kenya in the past year or so. So I thought that he would be, you might like to hear from him very briefly about this project. Okay, you have to hold that. Oh, you have to hold it.
There are two things I want to say first. One is sorghum, as Stan rightly put, is originated from East Africa. So my home is next to the center of origin of sorghum. And I don't just work on sorghum. I started working on sorghum before I was born. So the first working was by eating it because my mother was eating sorghum. And I grew up eating sorghum and growing sorghum, and I now work on sorghum. The second thing is that my country is Kenya, where we have made history by temporarily sacking the president. So, but we are working on that. Back to business. Stan said Halepens. He came here some years back, and he left his home and said he found a better home here. But it's not the issue really. I don't think it found a better home. It found problems. And because of the problems, hearing things like global warming and other things, it developed some very funny structures called rhizomes. And those rhizomes are what are making Stan sweat a little bit to make sure that things go right. So in Kenya, under this project, I am a sorghum breeder. I've worked on sorghum for the last 29 years plus in ICRISAT based in Nairobi. There are many other things we do other than the perennial sorghum, but under this project, we have received several populations, about 30 of them from Stan and Andy. We have put them out there. So the problems that Halepens, Johnson grass, as we call it here, went through and started adapting to mitigate against those problems are what we are now reintroducing to Africa to see how we can cope with the changing climate. So if we can get it working, then we are going to the right direction, because sorghum is resilient, that is true, but it's relative, because there's a limit, you can also push it. The climate is changing. The rainfall patterns are changing. The president of Tanzania, who is neighboring Kenya, just put the other day that the rainfall is becoming very clever, in quotes. So the farmers also need to be clever to plant the right crops to be able to get food. So if we can reintroduce what came out of Johnson grass as rhizomes and try to put perenniality in the sorghum and returnability, and then based on the erratic rainfall that we have in the region, then possibly we may be able to sustain food production, not only for human beings, but also for livestock, because we are talking of the whole cropping system and the ecosystem management per se. Also bear in mind that we have to sustain the system to be able to produce enough for the people to eat. So we have put out those materials out in the field the last season. They have over-seasoned, and the few that we have identified with the good returnability and rhizome formation, and with the time we intend to work with the team here and then introduce or integrate the trait back to adaptable farmer-preferred types in the region, and possibly now be able to tackle the problems of climate change and erratic rainfall, so that the farmer could be able to produce a crop even on very little moisture, because the crop will have established the root and it just needs little rainfall to be able to take off. So we look forward to more excitement as we proceed, and we hope that this will continue and possibly go into the second phase. Thanks so much.
We have a second project also working with Andrew Patterson that has been going on for during the same period as that. And we just have a minute or two, but I'd like for the two people doing all the work on this massive genetic mapping project, Wen Chen Kong and the Land Institute's Fiona Nabakalu, to tell you very briefly what they're doing.
Hey everyone, now I have the magic torch. So I started to work on the perennial sorghum since 2013 as a PhD student, and didn't realize that the projects makes me PhD duration a little bit more perennial than I would like it to be. So Fiona and I are about to finishing up and writing our papers. And as a geneticist, my daily work is pretty much like solving a jigsaw puzzles and understanding what the meanings of each jigsaw puzzle to all components of the sorghum plant. And in addition to that, as Eric mentioned, rhizomes, now I think 4 of us are becoming the world's best scientists to tell you the differences between rhizomes and the rest of the plants in sorghum. So with that, I would also want to emphasize that we're not only the dirt scientists, but we're also very cool manipulating data and computers and stuff, having robots in the field and taking the data and trying to develop new methods for high-resolution imaging so we don't have to work in the field anymore, so we can just stay in the air conditioning and play with the robot. And in the future, I would expecting there will be more robot grad students and postdocs. So they'll usually complain more, ask for more, and work more. I mean, complain less and work for more. Thanks.
Good afternoon, everyone. My name is Fiona Nabkalu, and I'm a postdoctorate fellow here. I remember my first time to attend the Prairie Festival. I told everyone I'll be around for 2 years for my postdoctorate, opportunity, but it's now coming to 4 years. So I think these plants have passed on the perennial trait in me, and I still think I'll be around for some more time. I would really want to continue being perennial in the perennial cropping system. So we've introduced perenniality from sorghum halepens into sorghum bicolor background through interspecific hybridization. That's how we get this progeny or the plant you're looking at. And among the goals of this project were to study the genetics of perenniality and also look at trait associations, and that's what we are working on with Wen Chen, Dr. Andrew Patterson, and Dr. Stan Cox. So from this work, we've been able to identify QTLs associated to different traits. And the other thing, too, we're interested in understanding the nature of associations between perenniality and other domestication traits, because people tend to fear that by breeding, progeny coming from Johnson grass into sorghum bicolor, we are likely to affect the domestication traits, or it might have some negative impact on sorghum bicolor or the domestication traits. So we want to know whether deleterious associations exist between perenniality and any other traits, because if such associations do exist, then that will hinder the breeding work. So we've been piling up lots of phenotypic data in the field, and Wen Chen has also been working a lot in the lab to generate the genotypic data. And based on the results we so far have, we are seeing that most of these traits show diverse genetic complexities with a pattern of at least one major gene and several other minor QTLs affecting these traits. And the environment also tends to influence QTL expression. We've also found that we've also not seen any highly significant negative associations between perenniality and other domestication traits. And that implies that we should not expect selection for perennial progeny or for perenniality to have any serious, deleterious, indirect, or negative effects on domestication traits. So breeding for perenniality won't affect other domestication traits. That's all I have to say. So, all right. So right now we are writing up most of these writings. We are developing manuscripts out of this work, and please look out for them. Anytime soon we'll be publishing, or they'll be in review. Thank you.
Who says there's no good news? All right, next we're going to invite Catherine Turner up to the stage. We're now in the lower level, as you might have noticed. It just keeps expanding. And Xuan Wang is about to finish his sabbatical time in China. He'll be back in the States, it looks like, October 10th. And we're greatly indebted to Catherine, who came on as a postdoc, who has really helped maintain the whole perennial wheat program here over this last year, as well as do her postdoctoral research. And so, Catherine, she has a few things to say.
So last year, about this time, our perennial wheat breeder, Xuan Wang, had just departed for China. And while Xuan was making new contacts and working on publications, our wheat breeding program here at the Land Institute had been scaled back a little bit. But we've still made some significant gains, and so I'm going to tell you a little bit about what our accomplishments have consisted of over the last year. So with a concerted effort, we made about 2/3 of the typical number of crosses from our breeding program. These crosses involve hybridizing one of several species of wheatgrass with either bred or durum wheat. And durum is the species of wheat used for making pasta. So we get the perenniality from the wheatgrass species, and then hopefully take some of the good baking qualities and seed size from the annual plants. So when making the cross between the species, it's not uncommon for the female plant to abort the embryo in response to foreign genetic material. So to prevent this abortion, we can rescue the developing embryo by dissecting it from the seed and placing it in a sterile environment with nutrients. The only problem with this method was that after John Mai's retirement last year and Shuen's departure, no one on our perennial wheat breeding group had rescued embryos. So like many times in the past, our greenhouse manager and our research technician stepped up to fill in the gap. So Tiffany and Adam successfully rescued hundreds of embryos last winter, and out of the many crosses that they made, there was only one misplaced pedigree. So not bad. Yeah. And we affectionately call that mystery line Little G for Adam Gorell. That's the G. So several years ago, Shuen devised a trial to test perennial wheat lines developed here at the Land Institute, as well as other institutions in a variety of environments around the world. Under the leadership of our colleagues in Australia, we're currently working on a publication to highlight the results of that trial. So what we found is that many of our lines perform much better abroad than they do in Salina due to a more moderate climate with more precipitation and cooler summers. So it seems like our first successful crops could be grown in other environments rather than in our own backyard. In fact, we have also begun sending seed by the kilogram to countries in the European Union for evaluation in large-scale trials that will compare soil improvements of perennial grain crops with annuals. But developing perennial wheat for Kansas in the Central Plains is still a high priority. So despite the challenge of extremely hot summers, we continue to find some promising individual plants. Last fall, Shuen selected our 14 best regrowing perennial wheat candidates. And over the summer, we harvested large plots of each of those candidates to test the quality of the grain for milling and baking. So we're going to be looking in collaboration with Kansas State for properties like protein and starch content, as well as the dough strength and elasticity to evaluate how the grain can be used. Our breeding program has typically used bread wheat as the annual parent in many of the hybridizations with wheatgrass, but we've started seeing a lot higher survival when we cross using durum wheat, the pasta wheat. So we think there are at least two important factors that enable stronger regrowth from the durum hybrids. The first thing is that durum has a smaller genome than bread wheat, so the overall amount of chromosomes and genetic material is lower in durum. And as a result, it is missing a bread wheat gene that controls the timing and temperature required for flowering. So without that bread wheat gene, we believe that the wheatgrass genes may be playing a larger role in the regrowth after flowering, enabling the plant to survive for a longer period of time. So that gene or pathway in bread wheat is still unknown, but we have a collaborator in Germany at the Max Planck Institute who's working on that project. So the other reason that the durum hybrids, we think that the durum hybrids survive better than the bread wheat hybrids is when we look at the genetics of the hybrids, we find that the durum hybrids have only lost a few wheatgrass chromosomes or even no wheatgrass chromosomes. And that's a stark contrast to the bread wheat hybrids that often retain less than half of the wheatgrass genetic material. So we see a correlation between the amount of genetic material from wheatgrass and the level of regrowth and perenniality. So by keeping more wheatgrass chromosomes, the durum plants tend to have stronger perenniality. So we've been using short DNA sequences to better characterize the genetics of the durum hybrids. And last year, I reported that the loss of the wheatgrass genetic material occurs kind of slowly over the generations in the durum population. And that relative stability in the early generations can be useful for releasing a variety in a short amount of time. So for example, we could make a hybrid cross in the first year and then self-pollinate for 2 to 3 years, evaluating those lines at different locations, and make seed available to producers in the 3rd or 4th generation, which would allow us to develop varieties in about 5 years instead of the typical scale, which is more like 8 to 10. So we'll need to evaluate more crosses to make sure that trend is consistent in different populations, but it's an exciting prospect. So besides the rate of chromosome loss, we're also finding out where the genes controlling key traits are located, much like the QTLs that Fiona was describing. So we have kind of a rough analysis so far, but we have found some wheatgrass chromosomes that are correlated with resistance to diseases in wheat, like powdery mildew, tan spot, and spot blotch. Those wheatgrass resistance genes can then be used to improve the hybrid crop. So we've also found a wheat chromosome that is important for the retention of wheatgrass chromosomes, and conversely, a wheatgrass chromosome important for keeping wheat chromosomes. So we need both the wheatgrass and the wheat chromosomes in the hybrid to have a viable crop. And we think that those retention factors are going to be important for maintaining a stable crop. So despite Shuen's remote location, we've still managed to make some promising gains. And to use Tim's love of the game as a metaphor, the progress on the wheat project is the product of a wonderful group of people who've taken on extra jobs to keep our bases covered. So I would especially like to thank Tiffany and Adam for adding embryo rescue to their skill set. Sheila, who took a break from sorghum and silphium to help with threshing, and Cindy for keeping a watchful eye on the plants in the greenhouse. Ron and John also helped with harvest this year, in addition to the normal crops that they work on. So looking forward to next year, I'm excited about leveraging some of our new findings and having everybody back on the same continent together. So, thank you.
Thank you, Catherine. We often boast that we rescue more embryos than anyone else in Kansas here at the Land Institute. So we're about to switch out of the grasses now and bring up a different plant family representative. David's representing the sunflower family, our oilseed crop work here at the Land Institute. David Van Tassel.
Thank you. And I work on a sunflower relative called silphium, as some of you probably already know. I remember reading an article in a magazine about how Boeing tests new jet engine designs. After years of refining the design, they build several engines, take them to the top of a mountain where there's very little oxygen, and then turn the engines up to top speed and let them run 24/7 for months to simulate years of flying. Do they overheat? Do they stall? What happens if you throw sacks of ice cubes into them to simulate hail? What happens if you dump dust and grit into the air intake as if they were flying through a dust storm? What if you toss in pillows to simulate hitting a flock of pigeons? Maybe they toss in sacks of dollar bills just for fun. I don't know. I don't think I read that, but they probably might as well, given how expensive all that is. Well, plant breeders are also supposed to do that. We're supposed to test our crops in very stressful conditions. But it's actually a lot more fun to breed plants with huge seeds or heads. On the other hand, it's terrible for farmers to grow a bumper crop and then have it topple over into the mud before it can be harvested. So we wait for strong winds and then look to see which plants in our breeding plots remain vertical. Thankfully, the Kansas winds never fail us. But what if the stems get damaged by one of many kinds of insects that enjoy burrowing in stems? And then the farmers get strong winds. And what if there's a drought? So how can we possibly bore holes in thousands of plants uniformly? If we released truckloads of common stem borers, the local farmers bearing pitchforks would run us out of town. Or we could, or maybe Wenchen could release swarms of nano-robots to systematically damage each plant. Or possibly we could genetically engineer and rear an army of insects that will feed only on silphium. Could we afford that? It's tremendously expensive. Well, I don't want to sound too boastful, but The Silphium staff has teamed up with an unlikely partner to solve this problem for pennies on the dollar. For years, we have secretly been creating the perfect conditions for Eucosma gigantea to flourish. This native Kansas moth feeds only on Silphium. It's so perfect. No other farmers will be threatened by our army of precision Silphium borers. They are programmed by thousands of years of evolution to seek out every last Silphium plant, no matter how well hidden. They know exactly what to do when they find one, and it's literally find and enter a Silphium plant or die. So they are absolutely single-minded and unstoppable. Fortunately, they are also 100% native, organic, non-GMO, and gluten-free. But it's even more beautiful. First, the moths lay an egg at night on each Silphium head. The caterpillar grows plump, eating out the seed head, providing us a huge savings on our insect chow bill. It then lowers itself to the ground on a silk thread and burrows into the Silphium stem. Almost no brain, and yet more precise than any nanobot. Note that we're also getting bonus stress testing by seeing which heads can handle being gnawed on by small germy mouths. Believe me, there are many other bad actors out there who can inflict similar damage on heads. Kelsey has seen weevils, wasps, flies, and other species of moths on the heads. Finally, you may be concerned that we will have to spend lots of money to keep our army of insects warm and fed over the winter. Fear not. We've again worked out a deal where they nestle down in the Silphium rhizome and feed on it all winter, and then emerge in the early summer, already perfectly located at each and every Silphium plant to repeat the process. So all of this is a long way of telling you that when you go on the tour and see that 80% of the Silphium stalks are laying on the ground, and if you look carefully, you'll see that most of the heads have been eaten from the inside out, don't feel pity. Don't see devastation. See success. See serious stress testing. See money in the bank. We're saving hundreds of thousands of dollars compared to building that swarm of nanobots or genetically engineering a Frankenborer. If a penny saved is a penny earned, Eucosma moth is one of the biggest single donors to the Silphium project. I've made a lot of poor assumptions about wild plants in the last 20 years. One of those is that wild plants will surely be super hardy and ultra-resistant. However, the average wild Silphium plant is actually very susceptible to Silphium rust and Eucosma moths. How can evolution have failed Silphium so badly? Well, for one thing, evolution is also constantly improving the nastiness of Silphium-specialized fungi and insects. For another, I have forced Silphium into situations with which it has no real history. Around here, drought and competition with prairie grasses means that Silphium patches don't get very tall or thick, seldom creating the stuffy, humid conditions perfect for fungal spores. And Silphium is sought after by rabbits, deer, cattle, and bison. The lovely towering Silphium plant growing in a botanical garden or prairie restoration is likely a historical anomaly. Probably for eons, most plants were grazed early and often. Escaping the bison long enough to flower may have been a rare event. Rare flowering would have kept moth populations low, as they need flowers to lay their eggs. The really good news is that there are some plants that seem to be able to have caterpillars drill through their heads without losing many seeds. And others have caterpillars in their stems, but by some miracle of design or sheer bulk, they stand strong and tall. There aren't many, maybe 1 out of every 200, but we don't need many to make rapid progress. Shouldn't we aim for zero tolerance of pests and diseases? Well, the cold virus is doing pretty well. Its fate is hitched to ours, and a stable evolutionary strategy is to outwit our immune systems, but nevertheless cause only mild symptoms. Breeding for total resistance to cold or Silphium pests may be an unstable solution or simply impossible. I believe that breeding a new generation of Silphium that gets infected but fights back to limit the growth of the pest and the Silphium plants that tolerate damage without falling apart is both possible and in fact already beginning. We now have Silphium plants with many small specks that show where a fungus spore landed and multiplied. But these same plants have strong, healthy leaves, and the specks stay small. Previously, the fungal colonies grew to great size, and the leaves turned yellow and wrinkly. And while many moth-eaten heads just seem to give up and turn to mush, possibly secondary infections, we do find a few that look clean and strong, if slightly scarred. I hope that you'll have a chance to look at the poster. It's on the wall out here, prepared by our visiting scholar, Luciana. She and her Argentinian colleagues have been comparing our highest-yielding silphium collections with wild cousins. There are some troubling signs that plant defenses may have started diminishing after several generations of easy living here on the research station. Well, the salad days are over, and it's the wild plants that are going to be having it easy from now on with my army of pests. I'm confident that we can reverse that trend of reducing the defenses. Given the range of stresses that our plants have been experiencing, I'm especially thankful that colleagues from many disciplines are now working on silphium and other perennial domestication. We need all the help we can get from entomology, physiology, ecology, microbiology, etc. We even have a small team of engineering students from Dordt College. They built us a unique seed wind tunnel last year, and the Matthews brothers, who you've probably seen out here on the cart stocking the outhouses, wrote some excellent code for it. This year, the Dordt students are modifying an old tractor to help us with the special challenges of weed control in a perennial. John Holmquist and his family are figuring out how to plant silphium and weed silphium using typical Kansas farm equipment, and a few days ago, the first production field was planted in the sunflower belt of Argentina. I keep trying to convince myself that the other breeders are secretly envious of my perfect storm of biological destruction as a mighty and priceless opportunity for testing and selection. In reality, I think that they have been quietly monitoring my progress through the 5 stages of grief. Most people move from denial to anger to bargaining with God. David is bargaining with moths. I confess that I wouldn't have minded having another 10 years to work on, say, the wings and wheels of the silphium jumbo jet before having to double down on making the engine survive hitting a flock of soaking wet ducks after being struck by lightning. But if nature builds you the perfect crucible and offers it for free, you'd better take advantage of it. Thank you.
All right. Thank you, David. Now we're going to have Tiffany Durer come up and tell us a little bit about a very special project that is unfolding right across the meadow here. So, Tiffany.
Hello. Okay, so as I was trying to figure out how to tell you guys about this last year, it has been so full for me of motivation and passion, and I am big into essential oils, and so my friend Laura is going to pass around some bottles. You guys are welcome to put them on, smell them, and feel the passion and motivation, because I got some exciting news to tell you about. You may have noticed on your Prairie Festival maps in the lower right-hand corner that there is greenhouse construction finally going on. And so, would you mind holding this for me, Lee? So I am going to walk you through what we have going on. Here is our science building, and as you walk out here through the fence, you got to keep the greenhouse people fenced off from the rest of the staff. You will see this area back here that is a great subject matter for elevation and how flat we originally thought this location was, but it is extremely not so much flat. So it's been built up quite a bit. So we now have this site work done for our new greenhouse. And, okay, we're gonna, upside down. I didn't. All right. Close enough. Okay, so I'm going to do a quick walkthrough. So this area over here in pink, this is something that I've been very motivated to get going. It is a new plant pathology greenhouse where they're going to be able to do experiments such as fusarium, which they have been doing in my greenhouse, right across from very healthy plants. And so I'm really excited for them to have their own space where they cannot cross-contaminate the plants that we've worked so hard to keep healthy. So that's going to be the plant pathology, and not only do we have that, but it is attached to a plant pathology lab. So we are currently in the process of interviewing and hiring a new plant protection ecologist, and this is going to be their space that is completely contained, so that way it cannot affect the rest. And then if we go up here in the corner, this is this outline area. This is the support house, or we call it the head house. And this is an LED room, and what that does is it gives us a place to put plants throughout the summer that are not quite as hardy and have trouble living through the summer in the greenhouse, or it just keeps them separate so we can do other types of experiments on them or start them. It just is a lot of flexibility that we really like to have here. And then this big green area right here, this is for the sorghum and the silphium. So I'm really excited about this. Because I am kind of tired of having silphium plants falling over because the shade cloth pushes them over because they're so tall. So these greenhouses are going to have 14-foot sidewalls, which should help contain some sorghum plants, but they'll still be getting exactly what they need. This is also going to be the silphium area, which I'm surprised David didn't mention because he is finally going to have some greenhouse space. Right now, he just gets to put his plants around everybody else's plants, and we kind of make do. So I'm really excited for him to have his own space where silphium plants should really grow and do really excellent. And then this down here, which I didn't do a very good job drawing this on here. Our greenhouse is too big for this piece of paper. So this yellow area here, this is the new legume area. And so Brandon will be up next speaking. And I'm really excited about this because it turns out legumes are really, really yummy, and every pest likes them. And so the whole greenhouse is going to have a bug screen to help keep pests out, and it should really help with the health of the legume project. So overall, it's been a very exciting and motivated year. I am obviously quite passionate about this project, and I look forward to next year being able to give you all a tour of our new facilities.
Thank you, Tiffany. All right, now we're going to move on to the last plant family that will be represented on the stage this afternoon. This is the Brandon plant family, or the legume plant family, actually, one that's dear to my heart. Brandon Schlautman is 1 year into his job, and we're going to find out what he's been up to. Brandon?
Well, thank you all for giving me the opportunity to speak to you again. Last year, I had been here only for 2 or 3 weeks, and so I kind of talked to you about ideas, visions, and dreams. And so this year, hopefully, I'll tell you a little bit about some experiences, actually. So, probably the best thing that happened this year in the legume program was hiring Spencer Barabalt to be the legume technician. He's been an excellent help and a great friend, someone to talk to about the decisions that I'm making and how to do things. This has been a year where we really learned how to manage all the different tasks that it takes for a legume breeding program. So thank you, Spencer. All the other scientists have been very helpful, sharing advice and ideas to help kind of guide me in this decision process. I'm trying to pick a legume species. Everyone else has kind of talked about the things that they've been, the particular crops that they're working on, and I'm still trying to find the crop that seems like it's got the best chance of becoming the perennial grain legume that we talk about at these future prairie festivals. 2 additional people have been a great help to me. Ron Kinkler and Adam Grail have been indispensable help in day-to-day activities, preparing the fields to be planted, getting equipment ready, and fixing things on the occasion that I might break them. So legumes, I want to remind everyone that biological nitrogen fixation through symbiosis with rhizobia is one of the defining features of legumes and their role that we're envisioning in our perennial crop assemblies. And by having this special ability to acquire this nutrient, legumes can hopefully supply themselves with the nitrogen they need to produce high protein, high quality seeds and forage. And possibly even to share that nitrogen with other perennial grains that they're grown in association with. So those are kind of the goals of the program. This first year has been a lot of imagination followed by trial and error. I spent most of the winter kind of imagining and envisioning what this perennial grain legume was going to look like, and then searching literature and herbarium specimens and working closely with Allison Miller and the global inventory team to try to decide what species to plant out here and to evaluate over the summer. And so my current task has really involved figuring out how to grow many different species, looking for which species have large seeds, have kind of croppiness type features to them, and starting to choose some to grow out here in Kansas. And so one of the specific characteristics that I really keyed in on and was looking for in plant species this year were legumes that had an erect growth habit with a terminal raceme. So they have this nice kind of raceme stalk with the flowers and stems on them that can be easy to harvest and is going to be above the green leafy material of the plant. And so one of these species that I planted out this year that kind of has that characteristics are lupins. And you might know lupins as kind of the ornamental flower that you've seen growing in gardens. They're very pretty. They have big, beautiful flowers on nice racemes that rise vertically from the center of the plant. And lupins have also been domesticated 4 different times. There's 4 annual lupin species that have been previously domesticated or consumed as grains by humans around the world. So we thought there was a good chance. And we have over 100 perennial lupin species here, native to North America, that we started evaluating. So this year, Spencer and I planted out more than 1,000 lupin transplants from more than 30 perennial species. And it turns out that not all lupins like to live in Kansas, or at least not at the Land Institute. A first wave of the species didn't like the many spring rains that we had and temporary flooding. A second wave didn't make it through the heat and the extended periods of drought. But a few species, particularly Lupinus leucophilus and Lupinus sericius, did very well, and they're still growing down on the farm. And so even though at the end of the field, end of the year when you walk through the field and there's lots of holes missing, this is a good thing, like David said. We performed the selection that we wanted to, and now we've narrowed down our list of candidate species to work on. So I'm excited to replant out those species that did well next year and start making selections. We also looked at alfalfa as a potential dual-purpose perennial grain, legume, and forage, kind of like we've been talking about doing with Kernza. And we planted out more than 1,500 alfalfa transplants this year. They were very hardy, did very well in the field, as we expected. It's a common crop. One of the best parts of alfalfa is that it's very broadly adapted. It can be grown near the equator and all the way into northern latitudes. It's probably the most bred, domesticated perennial herbaceous species in general that exists. And so with that, you get a lot of years of selection for very different resistance genes, traits, adaptation to multiple environments. There's a lot of researchers in the world that are already working on it, developing genetic resources, building genetic maps, sequencing genomes, et cetera. So there's a lot of resources to use. So I was very happy. Some of the plants that we planted out this year had really great seed yields. And so I'm excited we started digging up some plants and making selections, and it'll be neat to see how much progress we can make when we plant out the progeny next year. I'm also excited to tell you about some of the work that I've been doing with Tim Cruz and some other researchers around the U.S., looking at alfalfa as a companion legume, as a species that we plant with Kernza and our other perennial grains in order to supply nitrogen and other ecosystem services. And so this fall, this group of us have initiated a study planting alternating rows of alfalfa and Kernza, 25 different varieties of alfalfa, to try to understand what alfalfa characteristics are most compatible with Kernza. Do we want tall plants? Do we want short plants? Do we want plants with really big leaves, small leaves, and et cetera? And then also what alfalfa characteristics lead to high levels of nitrogen transfer. Alfalfa we know is great at nitrogen fixation, but we want some of that nitrogen to be transferred over to the Kernza grain crop. So hopefully this study, planted in multiple states, is going to help us to continue moving towards this vision of these perennial crop assemblies. This is one of the bicultures that Tim was talking about. So I mentioned that this year was a year of trial and error, and I came to know, like other researchers probably told you in the past, that domestication and development of a new crop species is much more than just a breeding and selection procedure. There's a lot of basic tasks that we need to learn how to accomplish. And so I took multiple trips visiting other breeders and spent a lot of time working with Spencer to figure out questions like, how do you thresh alfalfa seeds? How do you create crossing blocks and perform controlled pollinations between the species that we're looking at? How often do you water your plants in the greenhouse? What size of pot do you grow these wild legume species in, and with what type of soil media? So this is one of the things that I'm most thankful to Spencer for, because he understood that not all of our ideas were going to work every time, and he stuck with it, and together we worked out and solved many problems. And those solutions, that learned experience, is going to be kind of the foundation for many years working together to develop perennial leguminous crops. And so when we think about kind of an example of where I envision our program right now, Silphium and Kernza, a lot of these plants, we plant them and they grow a year without flowering and producing seed. They need some vernalization period. The first year they're kind of feeling out their environment and getting well established, and I feel like this was definitely a getting well established year. There's many interesting high throughput sequencing techniques and imaging technologies that have been mentioned, and I'm looking forward to using them someday. But this year we really learned a lot about how to grow plants well, how to measure the traits that we would eventually want to use in our breeding program. What tools do we need? We've acquired the right equipment to plant the crops in the field. So this year was a year well spent. I can imagine next year being a year where we're actually flowering, and I can't wait to see that happen. So thank you.
All right, thank you, Brandon. And we're approaching the end of this research update. If you've been here in the last couple of years, you will have seen Allison Miller, and she tends to wake up the audience. I think she might do that again today. So Allison, she's going to report on this global inventory that Brandon just referred to, and it's a project at St. Louis University in conjunction with the Missouri Botanical Garden, and is funded in part by the Perennial Agriculture project here. So Allison.
Thank you. Okay, good afternoon, everybody. This is another terrific Prairie Festival. I'm always so delighted to spend the last weekend of September here in Kansas. So as Tim mentioned, I am a professor of biology at St. Louis University and a research associate at the Missouri Botanical Garden. And for the past 20 months, I've been leading a project entitled the Global Inventory and Systematic Evaluation of Perennial Grain, Legume, and Oilseed Species. And the long and the short of what we're trying to do is to ask, what's next? We've heard of some incredible progress in a number of terrific crops, but we're wondering what's coming in behind that. And speaking with the theme of bridging divides, rural and urban, large and small farms, old and new farmers, we are bridging that divide between the breeding community based and ecological community based here at the Land Institute and the botanical community, the botanical research community. So I want to just share three major highlights of our progress, but before I do that, I wanted to introduce some special friends who are here with me today from St. Louis. We have a large team working on this project, so I want to just first recognize Claudia Ciotier. Claudia, can you stand up? Claudia is the postdoc on this project and has done an incredible amount of work. Great job. Sterling Heron. Sterling spent the summer last year at the Land Institute. He's probably dealing with his leg. Oh, Sterling, you're here. Okay, good. Thank you. Sterling is a legume enthusiast and a terrific contributor to the project. Heather Shear is a new graduate student at SLU, interested in nutrition. And then lastly, Maddie Ernest. Maddie is the director of the director of the GROW exhibit at the St. Louis Science Center, which has become involved in this project, which I'll mention in just a second. Okay, so the three highlights I'm going to talk about are related to the actual inventory, our work with legumes, and our work in education and outreach. Okay, so just briefly, the main goal of this project was to identify the number of perennial herbaceous species in the grass, legume, and sunflower family. We now know there are 7,566 perennial herbaceous grasses. I gave this estimate last year, but now we're sure. 6,656 perennial herbaceous legumes, and the Asteraceae is in progress. Sorry, David, we're still working on it. We've built a web-accessible, searchable database called Tropicos, where we didn't build Tropicos, but we built our project within that, where you can search through these taxa. We're populating that database with agriculturally and ecologically relevant trait data, and that the database will be searchable by those traits. I have a handout that explains the scientific details of this work, so if you're interested, please come see me, and I'll be happy to give that to you. That's the update on the inventory, just how many taxa there are in these families. For a number of reasons, we've been deepening our understanding of legume diversity, working closely with Brandon in a few key genera, including Astragalus, Lathyrus, Lupinus, Medicago, and Vicia, among others. We're developing some basic questions related to the biology of these species. So one of the things we're trying to understand is, aside from habit, perenniality, what are the major differences between perennial and annual members of the same genera? So Sterling's been leading up some germination trials, first at the Land Institute in 2016, in 2017 at the Missouri Botanical Garden, comparing seed size, germination rate, growth rate, flowering time, fruit production, soon root biomass and root architecture, to determine general differences between these two categories, annual and perennial plants, within the same genera. Okay, and then lastly, with regards to lupins, Claudia conducted a terrific series of field trips this summer to collect wild germplasm material of a species we're particularly interested in, Lupinus polyphyllus. So she spent time in Colorado, Idaho, and Oregon visiting native populations, collecting seeds, some of which have already made their way here to the Land Institute, and also leaves for population genomics analyses. So one of the things we're looking at moving forward is trying to bulk up the collection of materials that are available for breeders that come from native populations for some species that might otherwise have not been a focus of interest. Okay, so we talked about the inventory, the number of species, we talked about some work in legumes, and I just want to take a second to talk about outreach and education. So I mentioned the work of Sterling, Heron, and Heather. We've also been training undergraduates through this project. In the last 20 months, our lab has trained 7 undergraduate students in independent research projects in perennial plants and perennial crops and their wild relatives. We've also established a legume diversity display garden in the George Washington Carver Garden at the Missouri Botanical Garden with associated signage. And I mentioned Maddie earlier. So Maddie heads up this GROW exhibit at the St. Louis Science Center. And for those of you who don't know, this is the largest and I think first of its kind agricultural display at a major science center in the United States. The agricultural exhibit is in its just beginning its second year, and they had over 200,000 visitors last year. So Maddie was kind enough to allow us to plant Kernza, Silphium, and a Medicago and Alfalfa hybrid on display, and we've already been out there at least a couple of times talking with visitors about perennial polyculture agriculture and these crops. So in summary, the Global Inventory Project has been a productive venture that's expanding, we hope, the botanical foundation for perennial polyculture agriculture. I'd really like feedback from any of you. If you have thoughts or questions, please come see me, and thanks so much for your attention.
Thank you, Allison. Great stuff. And that does conclude the research update. I'm going to ask the researchers who will be on the plot tours to huddle out behind the barn briefly right after we're done here. And we look forward to seeing some of you out in the field. It'll be a little bit windy, and I'd bring a broad brim hat if you have one. And that those tours start at 3:45, or the first bus leaves at 3:45. Rachel was probably about to tell you this, but I'm jumping ahead. And if folks could be on time in those buses, that will really help this work. So thank you so much for your attention and your interest in our work, and we'll see you soon.